Hi, my name is Janie Munoz. Today I'll be presenting on exploring the Thioester intermediate capture strategy with Polyketide Synthase. So azinomycin. Azinomycin is a natural product that is produced by the microorganism Streptomyces sahachiroi. It was first discovered in 1954. However, in 1986, the structural determination was determined producing azinomycin A and azinomycin B, with the only difference between them is being the anneal substituent. This compound shows anti-tumor activities in vitro against lymphoma cells and in vivo against murine tumors in mice cells. It gains its anti-tumor properties from its abilities of DNA interstrand crosslink. So azinomycins are proposed as a hybrid PKS and NRPS secondary metabolites, where the naphthoate motility formation indicates a polyketide synthase PKS machinery, and the skeletal backbone is predicted to arise from the nonribosomal peptide synthase NRPS machinery. With these two biosynthetic pathways, the carrier proteins utilize a Thioester bond in order to bind to their substrates. So according to previous research, azinomycin biosynthetic gene cluster contains 5 NRPS and 1 PKS pathway. In our project, in order to study the azinomycin biosynthetic system, we intend to create a strategy to capture the intermediates involved in the PKS and NRPS pathway. The strategy was first developed by Dr. Washburn in Dr. Watanabe's lab. The theory of the strategy is using the design captain reagents to bind to the intermediates released from the PKS or NRPS enzyme. After systematically purification, we use LCMS to monitor the presence of the intermediate with the capturing agent. Biotin cysteine is the first generation of the capture agent, which has been validated in Dr. Washburn's research. In our project, we will create the second generation to see if it could be more effective and efficient. Instead of using cysteine as the amino acid moiety, the second generation capture agent doesn't contain the thiol group. We'll be using glycine, beta-alanine, and aminovaroic acid instead. By taking off the thiol group, the original proposed mechanism and reaction efficiency could be evaluated by using the PKS from the azinomycin biosynthesis pathway. So during the summer, we intended to start from 3 directions, which is to modify, synthesize, and prepare. So first, we optimize the scheme of the capturing agent. This is our overall revised synthesis scheme. We modified the reaction conditions as well as the purification methods. In doing so, we checked every step of this reaction by running a column chromatography, TLC, and NMR. First, we used a biotin ester and linker Bach to get the biotin linker Bach. Then we used TFA in order to release the protecting group to get the biotin linker. Next, we added the amino acid Bach to the biotin linker to get the biotin amino with the Bach group. And then finally, the last step is we use TFA and triethylsaline for the removal of the protecting group to gain the final product of the biotin amino acid. So second is we synthesize the capturing agents: biotin cysteine, biotin glycine, biotin beta-alanine, and biotin 5-ABA. So this is the first step. We use biotin ester and the linker Bach, reacted it in room temperature for 24 hours to get the biotin linker Bach group. Here, the NMR shows an H peak from the linker Bach moiety, indicating that the product is correct. Then we use TFA to react for 1 hour under room temperature to release the protecting group to get the biotin linker. The NMR indicates the Bach group has been removed. Bringing us to step number 3, where we use the amino acid Bach to the biotin linker with DMAP, EDC, and the solvent DMF. We ran the reaction for 24 hours at room temperature to give the product biotin amino acid with the Bach group. The NMR shows the protein peaks from cis-Bach moiety. So we did the same reaction with the similar purification process with glycine, beta-alanine, and 5-ABA. Currently, though, we have some impurities for the biotin glycine Bach and the biotin beta-alanine, which need repurification. The other two compounds, however, we have started the last step in removing the protecting group with TFA. As we came close to finishing the synthesis of the 4 capture agents, we started preparing the enzymes for the validation of the Thioester intermediate capture strategy. In our project, we used AziB, which is the only PKS from the azinomycin biosynthesis pathway, to test these 4 capturing agents. In order to get the halo AziB, which is needed to bind the substrate with the Thioester bond, we use ApoAziB with peptide SBB and coenzyme A. First, we made E. coli agar plates with PET24 AZB and PET21 SVP. Then we ran a mini prep, which confirmed that there's DNA inside the cell as shown on the gel. From there, we sent it to another lab in order for the sequence to be ran to confirm the DNA sequence. We then did overexpression of both of the proteins, which we then mixed them with coenzyme A in order to create the hollow AziB. As of right now, we're purifying the hollow AziB. Once finished, we're using it to mix with coenzyme A's in order to create the Thioester intermediate so we could start testing out the capturing reagents created. We collected fractions 7 through fraction 23 using 50 kilodalton centrifuge ultrafiltration for further use. In conclusion, we optimized the scheme of the capturing agents. We synthesized the biotin cis-Bach, biotin glycine Bach, biotin beta-alanine, and biotin 5-ABA Bach. Well, the final products are currently in progress. And finally, we prepared the hollow AziB to start the validation of the Thioester intermediate capture strategy. So I'd like to give thanks to the National Science Foundation, Dr. Watanabe, the Watanabe Group, the RU student researchers, and the organizers that put this all together. Thank you.